Ladies and gentlemen, esteemed participants and distinguished guests, I extend a warm welcome to all of you to today's online webinar on the environmental surveillance module in SORMAS. This is the state-of-the-art disease surveillance and outbreak management system. We're thrilled to have you join us as we delve into a very critical aspect of public health with focus on One Health, the environment. My name is Chinedu Arinze, and I'll be your host for today's session. And I also have the privilege of guiding you through our agenda and introducing our esteemed guest speakers. And we're looking forward to exploring the environmental surveillance module in SORMAS and how it plays a pivotal role in safeguarding the public health. Before we go into the details, I would like to introduce our distinct guest speakers for today. We're delighted to have with us Dr. Bernard Selenu from the Research Center for Infection, HZI, in Germany. And he will be giving us an overview of the science behind the environmental surveillance module. We also have with us Mate, who's a software engineer with the Vita Group. And he's going to be demonstrating the features of the environmental surveillance module, and he would walk us through its various sections. Right, and important to mention now that both speakers have very many years of experience working with and developing SORMAS. And we want to say a special thanks to both of you and to the HZI and Vita Group, also for the concept design and the development of this module. And we also want to use this opportunity to say a big thank you to the funders of the WAPA project who have made this possible. And once again, our dear audience, thank you for joining us. And without further ado, let's get started. I would start by allowing Dr. Selenu. Bernard, please, if you can already share your screen and take us through your slides. Thank you very much and welcome everyone.
Okay, thank you, Chinedu, and thanks everyone. And my name is Bernard, and as Chinedu has just presented, I've been working in SORMAS for some time now. So I am affiliated now with the Helmholtz Center of Infection Research. So the environment module in SORMAS, which is what brought us here, is an excerpt of a work pack of a project we are still doing at the Helmholtz Center. It's a water-based outbreak prediction in peri-urban Africa. We call it with the acronym WOPA. So I'll just give you a brief outline of the project before we go into the demonstration. So this will be the outline of our day. And before I start, I would like to appreciate or give some gratitude to all the partners. So it's not only us at HZI, it's a consortium, mainly of 4 partners and also a donor, the Grassroots University. And we have been working together. There are different components. So this presentation is only for a very small component, which is a work package 1. I will show now later. But before going to that, what was the motivation behind this? So the image on the left is just a simulated image on how it looks like in a real life scenario, where we find humans here and below, the 2 below circles, animals, which can be divided into wild and domestic. So these 3 entities always interact with each other, human, animals, and environment. And this is a definition from the OHITF report 2008, I just quoted so you see. And they said to attain a better health, we must consider all these 3 components as the definition for One Health. Then WHO also made a statement in their report, which was published here. And why I added it here is because there are some key components here which really tie to the vision of SORMAS, which is on prevention, detection, preparedness, response, and management. And that's really the concept we had when SORMAS was conceptualized. So we are moving forward in this direction. So far, we have done, let me say, a lot in the human part, but we haven't really done a lot in the animal and environment part, and this is the research we are going further. So I would just like to run you through this slide, how information flow through SORMAS. So on the left, you find a signal coming in, like an environment. You have a call that in a community, people are sick, it might be cholera or any outbreak, and you want to test to find out which are the pathogens in that environment. this is a case for a routine surveillance. And then the information will come into the system. And then somebody here we call surveillance officer. These are just personas. Somebody working at the district level can call him like the district surveillance officer. They are going to get that information documented in the system. And the technical word that Mateo will present later is called an environment. So an environment is just a place on the Earth surface where you collect a sample, but it's not on a body of a human or an animal. It can be water, it can be air, anything where you collect a sample for further testing. So we call it environment. So that will be collected, tested in the lab, and if there are other control measures, we have the control team who are going to do the necessary epidemiological measures that needs to be taken. So the outline of the work package 1 was to develop this environment module in SORMAS, and on the left are just some of the terms, some of the steps we envisage going through during the proposal, which involves a little rapid review of literature, specification, discussion with the users, and also with experts, because from SORMAS we develop involving everyone. And after that, we conceptualize and then program the feature. So the image on the right are other parts that are done, which is not part of this presentation, by other group members also involved in the project, but they mainly use the sample and then do further analysis in the lab. I will not talk on that. So for now, I would like to stop here after giving this overview, and I will hand over to Mate, and Mate will take us through how this really looks like in SORMAS. So thank you for your attention, and Mate.
Thank you. So obligatory question, do you see my screen?
Yes, please.
Wonderful.
Yes.
All right. So yeah, first of all, a small disclaimer. I'm not an epidemiologist myself. I'm a software developer, so things I present might not be 100% how it's done in the real life. And I also use primarily 2 users to do this stuff. It's not really a reflection of the workflow that Bernard talked about. So I'm going to use a lab user. I'm not going to report with an informant. This is all a bit toned down, because the purpose is to show you the different areas that we've worked on and that can be used in order to document all these things. So we're in the environment directory here, where I can create environments that then can be used in order to actually take samples from these environments and test those. And I'm currently logged in as an admin user, so someone who has access to basically everything in the system. So there's a lot of stuff here that's not relevant for our purposes today. I'm going to create a new environment now. And yeah, we have this screen coming up where I can select a few things. So first of all, an environment media. And what I'm going to select here is, we have different options: water, soil, rock, air, biota. I'm going to do a water media right now. And I'm going to call this Ölpersee because I have a system set up here that has some infrastructure data from Germany. And I'm living in Braunschweig, and there's this a lake here that is called exactly like this. So I'm going to create this as an environment right now. So let's see, this is in Niedersachsen, not in Berlin, in Niedersachsen, Braunschweig. And I also have some GPS coordinates that I'm going to put down here that are where this lake is actually situated at in real life. So 1 second. I'm also noting down the city and the postal code. Okay, so I'm gonna save this environment. And now I've been taken to the screen where I can edit this. I can add a few more things here. For example, the water type. So I have different options to put down which kind of water this is, which kind of water body. So right now this is a surface water, a lake. I'm going to select this. There's also the infrastructure details that I can put here. So again, this is surface water, but I could also document like water from a toilet or from a well here. So lots of different options. There's also the use of water. For example, whether this is like drinking water or something used for agriculture. Right now, I'm just going to select recreation because this is a lake that is primarily used for recreation purposes. And then I'm going to save this. And before I do anything else now, I want to show you another way of creating environments, and that's the import functionality that we have. So I'm going to open this. So we can not just like create environments through the application, but we can also import environments via CSV files. There's a whole import guide that I could download to have a look at that, how it's working. I'm going to skip that right now. I can download the import template, which I can fill in my data for, and then I can choose the file that I've prepared and use that to import data into the system. I'm going to do this right now. I have prepared a file with 4 environments. This is this one, and I'm going to start the data import. And as you can see, like all of them have been processed. Only 2 have been imported successfully. 1 has an error, and the other has been skipped as a duplicate. Because we also have duplicate detection for environments in the system. So what I've put down in that file was, among 3 others, an environment that is also the LPAZ. So it has the exact same details that I have just put down when I created the environment manually in the system. And SORMAS has properly detected this as a duplicate, so it did not import it because then we would have the same environment twice. I can then download the error report. And this error report will contain all the environments that I have that could not be imported correctly, and would tell me what the problem is. So for one of those, as I just said, it's a duplicate environment. So it would tell me which environment is the one that has been detected as the one that's already in the system. And for the other one, it would tell me that the environment media is missing. So in that import file I had, I didn't put down an environment media, so something like water or soil or rock. Showing this would be a bit complicated now. If this is like, if someone's interested, I can do this after the presentation and show you how this looks like. I would skip it for now. I can then use that error report file and correct the data and upload that again. It's this one. I've already prepared this as well. And then start the data import again. And as you can see now, we still have 1 skipped. This will stay the same because it is a duplicate environment, but the other one has been imported correctly now. I have just added the environment media air to that file. So as I said, I can show this later if anyone's interested. Okay. I will close this window now. And as you can see now, we have 4 environments compared to the one we had before. So these 3 environments have now been imported correctly through the import mechanism. And the last thing I will do with this user is I will again open the LPAZ environment, and I will create a task of the type sample collection. So I want someone to collect a sample of this environment. I will assign this to an environment user I have on the system, and I will save this task. And then I'm going to log out with this user and instead use an environment user to do the next steps. I'm going to quickly skip the dashboard because that's something I will show you in a minute. And as you can see, I have a lot less options on the main menu right now because the environment user I have in my system is really only supposed to work with environments in the samples. So as you can see, I have all these environments right now here that we've just created. And I can now open this environment. I see the task here, and now I will create a new sample for this environment. So I've now gone to the lake. I've collected a sample, and I will document this. So it's of course a water sample because I've taken it from a lake. Could also, of course, in theory, collect some soil from the lake. There's a lot of things I can put in here, like the volume, the turbidity, the temperature. I will leave all of this empty for now. We can also note down how the weather conditions were at the time of sampling, whether it has heavily rained in the past 24 hours. And again, I can put down the location. This has been taken over from the environment now. So when I create an environment sample from an environment, the location will just be taken over. But of course, if I, for example, collect samples from something that is as big as a lake can get, I might change the GPS coordinates around here a bit, because I might, for example, collect a sample from like the southeast of the lake, and then I want to create another or collect another sample from the northwest or so, because like there could be very big differences between those. I have to select the laboratory. Don't have one in the system right now. I will just select other. And then I can put down which pathogens I want to be tested. So this is optional. I don't have to do this, but if I'm already aware I want this to be tested for Vibrio cholerae, for example, then I can mark this. And I will also mark E. coli right now. This might not make sense in this specific situation, but just for demonstration purposes, I want to request 2 pathogens to be tested. I can also like note down whether the sample has already been sent, whether it has been received. This is usually something that the laboratory user would do, so I will just leave both of those things unchecked right now, and I will save the sample and then go into it. And now I can create pathogen tests for the sample. So I'm just going to do that. We're going to do that. There's a lot of stuff in here, like a lot of potential types of tests. Some of those are like primarily used for human samples. We'll come to this in a minute. So let's just select culture here as the type of test, and then I'm going to select E. coli as the tested pathogen. I can put down a date and time of results here. Don't have to do that. I will leave it out right now. I have put down something here. So if I would have selected an actual existing laboratory here, this field would not be present. And then I will just say negative. So I've tested this for E. coli, and the result has been negative. So no E. coli pathogens to be found in this sample. Then I'm going to save this and create another pathogen test, because I've of course also tested this for Vibrio cholerae, because this was requested. And let's just say this has been positive. So we actually have a positive test for Vibrio cholerae in this sample. So right now I have these 2 pathogen tests for the sample. And let's go back to the environments. And what I want to quickly show you now is if I go to the samples directory, as you can see on the top right, there's a switch here. I can have a look at human samples and at environment samples. This user I have set up has access to both. He does not have access to like the cases or contacts of the human samples, but can see the actual samples, because like this is an important part of One Health. So I have a cholera case in the system. I have a rabies contact in the system. And both of them are available to the user so that they have the overall picture. Because of course, if I like test an environment for stuff like cholera pathogen, then I'm also interested in whether there are actual human cases of cholera. You can see the sample that we've created right here. And now I'm going to take you to the dashboard. So right now, the only part of the sample dashboard that has been extended with the environment samples is the map. Everything else right now only shows data about the human samples. And this is, of course, something that can be extended in the future. I'm going to extend this sample status map. And as you can see, there's like, or maybe see, because it's very small, there's like a symbol here with a 3 on it. And if I click on this, then we can see 3 of these symbols here on the map in Braunschweig. And if I open the map key, again, this is pretty small on my screen, then you can see like red symbols are case samples, yellow symbols are contact samples, green symbols are environment samples. And this green symbol right here is the one that we've just created. You can see Ölpersee here. And this is basically like to the far southeast of the Ölpersee, of the lake, the sample that we have just created. So this is on the map. You can see exactly where this has been taken. If I quickly log out again for a second and log in with the admin user again, and just let me create an environment that is actually not in Braunschweig. So I'm just going to use something here. I'm going to use the Wannsee in Berlin, and I don't actually know where exactly this is in terms of GPS coordinates. And I also don't have the Wannsee district in my system even, but just for demonstration purposes, let's see. I need another window now. So let's put down some GPS coordinates that are actually in Berlin, but not exactly where the Wannsee is located. Okay, so let's save this. And I'm going to quickly create a sample for this. So this doesn't need to be, like, I don't need any actual data here. It's not necessary. Just create the sample. And then I'm going to go back to the dashboard, open the samples dashboard, and again, maximize the map. And if I click on this 4 again, you can see that there's another sample in Berlin here. You can see the symbol there, right? So that's the one we just created. It's pretty far away from the Wannsee, actually. I think the Wannsee is somewhere this area, if I'm not totally mistaken. And now I'm going to log back in with the environment user. And this environment user is a user that is restricted to Braunschweig because like this is their area of operation. And if I open this dashboard again, you can, and I like go over to Berlin, you can see there's no symbol there. I can't see that because it's not in my jurisdiction. And also, if I go to the environments directory, this environment, this Wannsee environment isn't even showing here. So I don't have anything to do with that. I don't see that data because it's outside of my jurisdiction. All right. And now I'm going to switch over to the mobile app. So I'm going to quickly stop sharing my screen because I need to switch over. Let's see. All right. Quick thumbs up from you, Chinedu, if you can see it.
Yes, please. We're good.
Great. Amazing. All right. So now I'm in the mobile app and I'm logged in with the same user that I've just used, the environment user, not the admin. And so right now you can see there's no environment there because I've just I've opened the application before I created all this data in the web app. So first thing I'm going to do is to synchronize, and you will see some fragments, like some disturbances on the screen. Those can be pretty bad at some point. This is not the app, it's the emulator I'm using. So SORMAS is flicker free. So just like bear with me through these annoyances. So synchronization is complete. And as you can see, we have those 4 environments here on this app that we've just created in the web app. Among them, the ÖPAZ. So basically, the mobile app is a recreation of what I've just shown you in the web app. Like all the fields that I've shown you are here as well. I can create environments, I can edit them, I can create samples for them, I can create pathogen tests for them. I'm going to quickly show you that, but I'm not going to like create new data right now because it's essentially more of the same, but just like working in the mobile app as well. So this is the ÖPAZ environment that we've created, including the location. And this is in read view, so I can also go to the edit view, and now I can also edit all this. For example, I could add an external ID right now, and save this, and then the external ID would be added. You can see it right here at the top, 1, 2, 3, 4, 5. I can also go to the environment samples and see the sample that we have created in the web app as well. I could create a new sample here, and I'm going to do that right now. So I'm going to create a new environment sample for the ÖPAZ because, for example, I want to use a new location right now and collect some water there. So again, I'm going to like select water here as the sample material. The data is automatically filled in the mobile app. All these fields that I showed you, like the temperature, for example, are here as well. And also the location. And the location in the mobile app is automatically picked based on the GPS coordinates of the device. Unfortunately, in the emulator, this doesn't properly work. I cannot send... These disturbances are pretty bad right now. I cannot properly send the GPS coordinates of an actual location at the ÖPAZ right now. So this is like just like standard coordinates that have been put into the emulator by Android Studio, by the software I'm using to develop the application. We're just gonna have to deal with those. So ideally, I would like to change them. I could like click on pick current GPS for address, and then it would actually work. We're just gonna use those. I have to select the laboratory as well again. And then I'm going to save this sample without requesting a specific pathogen test. So you can see the sample is here now. I could also create a pathogen test for it, just like in the web app. I'm going to skip that. And then I'm going to go back to the environment listing and I'm going to synchronize. And now we have to go back to the web app again. So again, quickly have to switch what I'm showing. 1 second. And we're back. So
good.
Yeah, great. I'm going to go back to the ÖPASee environment. And as you can see now, we have 2 samples here. And the second one is the one that we have just created in the mobile app. And if I go to the dashboard, you can see we have a brand new sample somewhere in the US, which is these GPS coordinates that have been automatically put in there by the software I'm using. It should be somewhere in San Francisco. But it's there. So in a real life scenario, you would have actually put down a proper location at the ÖPASee and you would have a second sample icon showing up at that lake. So let's just pretend that this is the ÖPASee and this is a proper sample there. So as I said, that's just my emulator not working correctly in terms of GPS data. And yeah, I think that's everything I wanted to show. If there's something I've been missing for some reason, Chinedu, Bernard, please let me know. I can do that. But otherwise, I would hand back.
Thank you very much, Matej. This is well appreciated. Yes, and very detailed as well, taking us through the web and also through the mobile. I think that was very important that we showed and that people understand that this application also works on the mobile for people who are even in poor resource setting areas where there are very limited internet connectivity. They can already collect data and then synchronize when they have access, and that information is on the server for further use. Thank you very much, and do have a pleasant day.